Burden reference. Okay, so thank you for coming today, those of you that did. Our presentation is going to be on demystifying EEG. We're going to go over the research that I'm conducting in my lab, some data and analysis, and what the future holds. So my name is Kyle Giffen. I'm on the publications team in neurotech, and I've been in here for 1 semester, and I'm studying cognitive science and data science.
Hi, I'm Connor Kennedy. I'm in the software team. I've been here now 2 semesters, I guess, and very excited.
All right, so without further ado, let's get started. The lecture outline, we're going to go over the discovery of EEG, which I think is a really cool story. And then we'll talk about what it is, how it works, and why it works. Connor's going to tell us where we place the electrodes and label them. We'll go over some research data and how to clean it, and then what we can actually do with EEG. So to start the discovery, in 1780, this guy, Luigi Galvani, was showing all his homies a pretty cool trick, which was that if he connected a generator of static electricity to the legs of a recently dead frog, seen here, the frog's legs would actually twitch, despite the fact that it was dead. Galvani was surprised by this, and he dubbed this animal electricity. And he postulated that every single animal, every living animal, has electrical currents that run through it. People thought he was crazy, but over 100 years later, this guy discovered that the brains of rabbits and dogs also exhibit electrical activity. So 34 years later, we have Hans, and Hans Berger, he dropped out of school after studying physiology and went to the cavalry and enlisted in the cavalry. And one day in the cavalry, he's riding on his horse, he gets knocked off, and another horse carrying an artillery cannon screeches to a halt and nearly tramples him over, nearly kills him. And so he goes back to the barracks not 5 minutes later, and he's received a telegram from his sister. And the telegram says, "Hans, be careful. I sense that you're in extreme danger." Because of this bizarre coincidence, he became obsessed with telepathy. And oddly enough, he created this device, this elaborate device right here, that is now known as the invention of the EEG. He was the first person to ever record a signal from a human brain. So if we fast forward to now, the EEG is used for research and medical purposes across the world, allowing people to better understand how the human brain works. And so now I'll pass it off to Connor to tell us more about EEG.
So, as I'm sure you all know, the human brain is made up of billions of neurons sending each other electrical impulses. And so what an EEG device does is it picks up on these tiny electrical signals and is able to measure them in real time. And so it does this using electrodes placed on the scalp of your brain, and these measure slight variations in the flow of electricity. And so it creates this graph, as you can see in the bottom, that basically measures the electrical signal at that electrode in comparison to some other reference point on your brain.
Question?
And so there are many actually other ways of looking at brain activity as well, such as fMRI, PET scans, CAT scans, and NSRI. And these all have really high spatial resolution. So they allow us to look at very specific areas in the brain and see what's going on. But what they don't have is high temporal resolution. So this is what leads us to EEG.
Before I proceed, Alfredo, do you have a question about anything?
Yeah, so you know how the brain has chemical synapses and electrical synapses? Does EEG detect both, or is it based mainly on the electrical ones?
No. Connor, go ahead.
Sure. So chemical synapses, they create electrical impulses. So the chemicals flow, charged ions flow from one side to the other, and they create an electrical signal in the brain. So they're actually all creating electrical signals, really. It's just some of them
both
use charged ions to do that.
Thank you.
So as Connor mentioned, all these other neuroimaging methods give us really good spatial resolution, which is this beautiful image of what the brain actually looks like, intricate detail of all the minute structures. But what we're not getting is when this happens. And that brings us to why EEG is so special, is it has really high temporal resolution. So this is that ERP, the event-related potential that Connor was just showing you. You can see from 0 to 1,000, these are in milliseconds. This is just 1 second of information. We can see a hyperpolarization and then depolarization, and these small fluctuations in neural activity that happen at very, very small time frames. This is pretty unique to EEG. Again, down here, 10 electrodes, each with a different signal being generated, and micro fluctuations at milliseconds of time. So this is amazing for telling us when things happen. Another awesome aspect of EEG is the spatial variation. So not only can we record a single neuron by placing an electrode next to it, we can actually record a cluster of neurons and a cortical layer, and then a large area of brain, which is what the EEG does. And by comparing the signals generated at different spatial variations, we can get an understanding of what happens at the macro and micro level. Another cool thing is species variation. So even honeybees have little central processing units, little tiny brains. And oddly enough, the temporal activity of their brains very much mimics that of a human. And I meant to have a human face up here, not an ape, but anyways. So we can study in honeybees and then rats and cats and humans, and we can get an interspecies idea of how neural activity is generated and basically how cognition is generated. And then finally, it just looks super cool. I mean, look how happy and dope this guy is on the right. So these are kind of what makes EEG special, and I'll pass it off to Connor to go over electrode labeling and placement.
Absolutely. So originally when EEG started being studied, every experiment would have their own electrode placement. So there was no standardized system for what was going on. And so it was really hard to compare across studies. So eventually this international standard was introduced called the 1020 system of electrode placement. And so that is what you're seeing right here. And so it has both the labeling of the electrodes and their placement. So on the left side of the brain, they are numbered with the odd numbers, 1, 3, 5, etc. And then on the right side, they are labeled with the even numbers, so 2, 4, 6. And then each of the letters corresponds to the area of the brain that it is in. So FP is prefrontal, F is frontal, then central, temporal, parietal, and occipital. And so, and then you also see some electrodes labeled with 2 different letters, and these correspond to areas in the middle. And then another important area is the so-called 0 sites. And so these are on the center line of the brain, and they hover over the corpus callosum. And they're really used as reference electrodes, right? Because there's no brain there, like directly in the middle. And so you compare the electrodes on both the left and the right to what's going on in the middle. And then they're also numbered such that as the numbers increase, you're moving out on the brain. So this makes it really easy when you're looking at an electrode to keep track of where it is on the brain.
Okay, so that brings me to the actual research that I do in my lab. So we put this funny hat on you that makes you look like a clown. And I'm actually doing this to our patients. I'm putting this substance in their scalp, this conducive electrode gel. And it looks like I'm just jamming a needle into their head, but really I'm very carefully, the needle is dull, and I'm carefully moving their hair out of the way and then just ejecting a little bit of gel under each one of these holes. And then making, small talk while I'm doing this, which is always super fun. And what we want to do there is to take these biosemi electrodes, each one labeled differently corresponding to function and distance from the midline, and place them into the scalp such that we have the best possible connection. So we tell the electrode, go to your home. The electrode goes home. And then we do this 64 times. And I want to mention that these ones are the facial electrodes. So these will actually be placed under the eye and on the cheeks. And all of these are not all named FP1. I just used that image for all these. But this is what that looks like. So this is actually my head researcher, Beth. And Beth is wearing an EEG electrode cap. And you can see that midline that Connor was just describing in the different labeling. And she's choosing between two shapes on this screen. And this is part of our research studying reinforcement learning based on whether people are motivated by points or goals. I'm not going to get too far into that, but I just want to give you an idea of what the setup looks like. So this brings us to understanding actual EEG data. And I'm going to stop screen share and have Connor pick it up because he can display the video better for us.
Here we go.
All right, so play that one more time. Perfect. So this is actually data being generated live in my lab. And Beth is wearing the cap, as you saw in the previous slide. She's intentionally causing a lot of noise. So this is not a clean signal. You can see the noise, like she's blinking rapidly. She's doing all these things to mess up the signal so that I can see what it looks like. And then on the right video, she's going to move her eyes to
the EEG signal that isn't brain is horizontal eye movement. The EEG signal that
Sorry.
No worries, it's perfect.
isn't brain is horizontal eye movement. So if you can look all the way to the left and all the way to the right a few times. Right, so you see these plateau looking things? Those are your horizontal eye movements.
Okay, cool. And then Connor, if you want to stop share, I'll pick it back up. Okay, so can you all see my screen? Yep. Okay, I'll take that as a yes. So this is a really important part of EEG, which brings us to the data that we want versus the data that we don't want. Over here are different frequencies of neural activity that have been studied and found to correspond to different cognitive states. So this is real neural activity. Delta, very slow, low frequency, high amplitude would be a sleepy dreaming state, whereas alpha is more relaxed and reflective. So if we look at this data to the right, and Connor taught you about the electrode labeling, you should be an expert on this now. FP2 right here is the prefrontal, and that would be on the right side. And FP2 shows this kind of alpha activity. And amongst like all these waves, there's real neural activity that we want to decipher. But in between these, high amplitude, annoying blinks. And so how do we remove these? That's what we're going to get to. These are called artifacts, or as some refer, the Carol Baskin of EEG data. They come in and they disrupt everything and cause a bunch of chaos. And so here's what we have to do. We have to isolate these artifacts. Actually, I'll do a little demonstration right here. If you put your fingers to the side of your temples, like so, use mirror neurons and mimic me right now. Connor, you too. I'm looking at you. And then clench your jaw like this. You'll feel muscle activity occurring in your temples. And this occurs all over your head. So all the electrodes are noticing this muscle activity and picking it up. And it causes massive distortions in the signal that we're generating. Another thing is eye blinks. So eye blinks, again, we have electrodes on the face, AF8, AF4, AFZ. You're seeing these high amplitude eye blinks. We want to get these out of our data so that we can isolate the neural signal. And then lastly, we have horizontal eye movements, which is what Beth was telling us about in the video. These kind of plateau looking data points. So amongst all the noise, this is the good data. And how do we isolate it? Well, we could just delete all this part, and that's called epoch rejection. So part of my research is actually going and finding noisy parts and literally deleting it. But that still eliminates some of the good neural data. So if I delete it and we're just left with this, maybe there's some information in these epochs, like right here, that is clean that we want. So we could just delete it, but there's a better way. The better way is individual component analysis. I found that the best way to explain this is to use the analogy of the cocktail party effect. So imagine you're at a cocktail party, as we all frequently are, and you are talking to a person next to you, but there's a crowd behind that person, and there's people chattering, and there's a bartender clinking glasses, and there's music playing. How do you isolate just the person you're talking to and filter out just that range of stimulus? So here's another visualization. Follow me here, because this is important. We have two speakers, each speaking at different wavelengths. And then we have a crowd, and we have two microphones. So this microphone is going to pick up information from this speaker, and it's going to pick up information from this speaker, and it's going to pick up information from the crowd. So will this microphone. Speaker 1, speaker 2, the crowd. And then it combines all this information it's getting into one signal. But if we feed these signals in that contain both speakers and the noise from the crowd, we can run it through ICA and actually recover the information just from speaker 1 and just from speaker 2 and eliminate the crowd noise. So if you think about this in terms of the EEG, the speakers are neural signals. Our microphones are electrodes, and the crowd is our noise. The crowd are our artifacts, our blinks, our jaw movement that is contributing to this wrong signal. So if we run these through ICA, we can recover the data. Before, we might have all this noise, this high amplitude jaw, blink, and saccade activity. But after ICA, we can flatten and smooth out the data just to get the neural signals that we want. And this is an extremely, extremely powerful algorithm for analysis. So to get into this real quick, the algorithm runs over the data and sorts from all 64 electrodes the patterns of activity by variance. So it's important to note that there's no jaw activity in this analysis. I didn't run this analysis on a data set with jaw activity, but the most variant are these eye blinks. So you can see that ICA actually sorted the most variant activity, which is these eye blinks, and then saccades right here. It then plots this activity for me to look at, analyze, and reject. So this number 1 high variant signal corresponds to this plot, this topographical plot. The colors you're seeing here, red is for high amplitude and blue is for low amplitude. So if I select this plot, I'll select this component and analyze it. We can actually see that, okay, it's mostly occurring at the front of the face and near the prefrontal area. And we're seeing these high amplitude, consistent marks of data. Well, that tells me these are probably blinks. And there's a lot more analysis you could do, like look at the power spectrum and look at the ERP response, but we won't get super into that. But this is basically to show that you can select patterns of activity, eye blinks, eye movements, and reject it. I mark this for rejection. And now when we look back at our data, it's no longer there. So ICA took care of all the annoying stuff, and that was a significant benefit to us. So this is the neural data that we're interested in, and I'll pass it off to Connor. Connor, do you want to switch screen sharing right now?
Yeah.
Question?
Before we do that, Alfredo, it looks like you had a question.
Yeah. What do you need to get ICA to work? Like, is this an algorithm that we already you just run and it knows what it has to eliminate, or do you need to input it something apart from the data?
So, Connor, if you go one slide to the right, that one, yeah. ICA. Oh, I forgot to mention this, but this whole window of data you're seeing on the left is only 15 seconds of information. So imagine 20 minutes. This would be many, many windows of data, right? Okay. Is an algorithm that will run over this data and train and learn what are the most high variance patterns of activity and how can we sort those so that the researcher can identify those patterns and see if they're worth rejecting. And so that's what it does for us.
Yeah, and here's where you choose whether to reject kind of a section. So
okay. So ICA runs manually, but then you choose, it just gives you some options and you choose what to reject.
Yeah.
Okay.
Okay, so now that we have this clean data, we're really looking for two different things, depending on what you're researching. So either event-related potentials or evoked potentials. So event-related potentials, or ERPs, are essentially things that you are doing. So motor movement, thoughts, anything. Evoked potentials are just things being done to you, such as a stimulus. And so once we discover what these are, we can actually analyze the data using many different methods, such as Fourier transform, spectral analysis, and a few others. to allow us kind of to match what ERP corresponds to like what is actually happening in the real world. And so this allows us to create models for computation where we can look at the brain data and actually understand, okay, what is really happening? What are you thinking? What part of your body are you moving, et cetera? And so this is really powerful and allows us to do some really cool things. So here is one example.
A quadcopter flies through a balloon ring. Cool, but what's the big deal? Well, look, Mom, no hands. There's no mouse or joystick here. It's all the power of thoughts.
Then the signal coming from his brain is being picked up by these sensors and then decoded and sent through a Wi-Fi system to control this flying quadcopter.
With support from the National Science Foundation, biomedical engineer Ben He and his team at the University of Minnesota created this brain-computer interface to help people with disabilities regain the ability to do everyday tasks.
The goal is eventually to make this technology to go to really help people, say, on a wheelchair with a variety of disability.
Professor He is still testing out the system with college students. Before maneuvering the quadcopter for real, they train for 10 to 20 hours using a virtual aircraft flying over a computer-generated campus.
So that is a super cool example of, you know, one thing that can be done. And he explains a little bit later in the video that he controls the quadcopter by thinking about either throwing a ball with his right hand or catching it with his left. And so that is some of the directional control. And then another example of what can be done is right here.
The goal of our research collaboration is to restore function to people with paralysis. This can be people who have a spinal cord injury, people who have a neurodegenerative disease like Lou Gehrig's disease.
We're able to eavesdrop in on electrical activity and slide a cursor across a keyboard and type out messages.
Our research is focused on an area called the motor cortex. The motor cortex is responsible for generating movement on the opposite side of the body. Even in cases where you have paralysis, those cells, those neurons in that area of the brain are still active.
We can surgically implant a tiny electrical sensor that's made out of silicon that has 100 tiny electrodes that just sits on the surface of the brain, and it's able to pick up on the electrical activity of individual brain cells or neurons.
We record tiny signals from those neurons, those brain cells, called action potentials. We can then take those signals and decode them using a computer. We can manipulate them once they're in digital form like any other data.
So all you need to do is imagine moving your right arm, for example, to the letter T on a keyboard, and it'll slide out to the T. And
so as you can see, this is an example of actually a more invasive method where they actually implant something in the brain, but the functionality is very similar. So, and is a great example of what could be done with EEG. Do you want to start sharing again?
Sorry about that. I was muted. Okay. Can you guys see my screen? Yeah. Okay. So taking it from here, let's take a quick look at some of the commercial EEG companies that are out there today. If you think of this as like the Verizon flip phone, what will be the iPhone X? Honestly, like these are just very basic EEG devices. Muse is used for, you know, meditation, Halo for exercise. These, OpenBCI and Emotive, they're allowing people to do individual research on their own brains. Dream is allowing people to analyze their sleep patterns and optimize their sleep. And then Neurosity is trying to get people into a flow state with coding. These are just some of the commercial EEG devices that are out there today you're probably familiar with. The applications, though, are incredible in that we can enable people with disabilities, even in the short term, as you saw with that woman studying with the Stanford researchers. We can help people with paralysis, people who are in wheelchairs. This guy can actually control his wheelchair using his thoughts, the movements of his wheelchair. This guy is being rehabilitated to walk again, and the EEG is analyzing his neural activity. So what we can actually do like now is slowly send messages, control light switches, open and close doors, potentially adjust a thermostat, things that are kind of smart tech, like easy binary responses you could indicate with thoughts and intentions. And in the long term, you know, hands-free computer navigation, like you saw with that woman, rapid thought to text. Facebook is investing a lot in this. They plan to be able to do 100 words per minute from thought to text. Remote flight control of drones, or even of real airplanes, if you could fly an airplane remotely. Virtual reality and gaming, no longer using controllers like Xbox or PS3 controllers, using your thoughts to guide you through the experience, could potentially integrate with self-driving, and I imagine could have wide commercial availability. And there's several huge players like Kernel with $100 million in funding, Neuralink, $100 million in funding, that are kind of pushing the boundaries of what we can do right now. This is Neuralink's model of having a directly implantable BCI that goes underneath the brain. So when we're talking about EEG right here, this is very non-invasive, but this would be a directly invasive approach where you insert into the brain. And then lastly, Control Labs up here was just bought out by Facebook for $1 billion. And they allow you to send signals through your wrist and to control outcomes on a computer through the intentions of motor movements that you send through your wrist. So the applications are very, very wide ranging. Yeah, Alfredo.
Do you have any idea on what the state of the art for like maximum axis of movement is? So like a cursor would take like 2 axes of movement, like up, down, left, right, and like typing would take like, well, at least 27 for characters. So like, do you know where we're at currently?
Interesting. Would you imagine a 3-dimensional axis of movement such that you could choose to move things forward and backwards, or?
Yeah, so that would be 3, right?
Yeah.
And then like 4, we can't imagine it physically, but it'd be like choosing between 4 things, and then like between 5, like 5 actions.
The drone is 3, at least. So the majority of the research goes into prosthetics. So that's where almost, like if you talk about all the research funding, so there might be a few, maybe typing or a few commercial options, but really it's going to be mostly in 3D space because that's where all the research is, or a lot of it.
That actually brings us just to here, which that was it. So thank you all for listening. And yeah, if you have more questions, it would be interesting to talk about. Go back to anything.